Let's start simply, you guys. Others may join us, but I always like to keep this efficient. So genomics, you guys, the study of an organism's complete set of DNA, and we'll be primarily referring to the human genome. So let's talk about DNA. DNA is the language of life, and many of you will probably be familiar with this at some point in your education. And that is DNA is divided into or packaged into a number of large sequences that we call chromosomes. And in the nucleus of nearly every human cell, there are 23 pairs of chromosomes. Okay, 46 in total, one set from your mom and one set from your dad. Now, these chromosomes typically are numbered from 1 to 22. And most of this, you have to remember. Again, I just want you to have a mental model. You'll see as we get to the end. But most of these chromosomes are numbered from 1 to 22, typically from largest to smallest, and then followed by a pair of sex chromosomes at the end. Okay, and one full set of chromosomes from an organism is called its genome. Okay, and this contains all the information needed to create you or any living organism. That's all the information we need, and that's what we study when we're looking at genomics or bioinformatics. You're going to see what I mean as we dig in deeper. That's what we want to get. Okay, that's what we sequence, and that's what you're studying. That's what you're analyzing. Okay, so inside a given cell, nearly all cells, I'm talking mainly about the human genome, you're going to have, remember, 23 pairs of chromosomes. And each chromosome is a tightly wound bundle of DNA. So it's a sequence of DNA. That's where your DNA is. And remember, we talked about those 23 different chromosomes. That's where your DNA is. It's just a sequence of DNA. And I'm going to break this down for you as we go here. Guys, let me see here. If you have a question, guys, feel free just to unmute. This doesn't have to be a monologue. I'll keep my eye on. Hey, Malika, I'm so glad you're here. And feel free to put something in the chat if you don't want to speak up. And I'll try to see it. If not, I'll certainly get to it as we get toward the end. Now, when we talk about DNA, you guys, deoxyribonucleic acid, or DNA, the information in DNA is stored as a code of 4 chemical bases, which you may be familiar with, or nucleotides. We'll also call them nucleotides, which are slightly different. But bases of nucleotides, we're going to use interchangeably here. Okay, so we have adenine, thymine, guanine, and cytosine. You don't have to remember those, but I do think it's helpful to know these letters, A, T, C, and G. And these are base pairs that A and T can only go together and G's and C's can only go together. We won't talk much about that, but you'll see how that's helpful as we go forward in terms of understanding DNA, okay? Now, it's these sequences here? This is normally how we see DNA, kind of the stair-step ladder. These sequences of bases, okay, those are purposeful. It's the sequence of these bases that determines the information for building and maintaining an organism, okay? And again, like if you don't have much biology or molecular, don't worry about it. We're going somewhere. Okay, so just remember a mental model. We're going somewhere. Now, again, inside virtually every cell, 23 pairs of chromosomes. Inside a chromosome, you have this tightly wound DNA. You have a sequence of DNA. And a particular segment of that DNA, you guys, that produces that produces protein, we call that a gene. A gene is the segment that contains instruction for making proteins. Okay, and you'll see that that's very important. That's where where most of the protein producing instructions are located within genes. It's that part of the DNA that has instructions for creating the things that we need to make an organism. Okay, so this is the first take home I want you to understand. A chromosome is what holds your DNA. And a segment of that DNA on that chromosome is a gene, and it has the instruction to create the proteins to keep the cell alive and executing its specific functions and all of that. Okay? So the first part, here's the first part. If you've gotten everything up to this point, you should get this. Let's summarize what we've said up to this part. We said within a cell, nearly every cell in the human body, we have 23 pairs of chromosomes. Within a chromosome, we have a sequence of DNA. And within DNA, we have a segment of DNA that codes for a variety of proteins. That's called a gene. Okay, let me give you a mental model for DNA here. A mental model for DNA is this. You can think of DNA, you guys, as telling a story. It's like reading a book. It's your biography or whatever. It's this story written using only a 4-letter alphabet. That's what I think of it. It's just incredible to me. It's this 4-letter alphabet, A, C, G, and T, representing, remember, bases or nucleotides, if you want to use that language, or you could say letters, whatever it is, or bases is commonly used. But it's this 4-letter alphabet. This is the genetic code, this 4-letter alphabet. And conceptually, you can think of DNA as just a long string of A's, C's, G's, and T's describing you. or any living organism, whatever it is, the thing that makes your hair hang that way, or that dimple, or the way you walk, or the way maybe even part of your personality, and all these different things is this story that's told in this four-letter alphabet. Okay. Now, a segment of this alphabet, a certain part of this alphabet, a subsequence within your genome or DNA. Remember, DNA genome, which is all of your DNA, all 23 base pairs, your genome or your DNA. A subsequence of your DNA, we call a genomic sequence. Okay. And it's genomic sequences that we're going to be analyzing for their story and that information. Tell me about this and that person. Why this, or how will they respond to that? Whether it has to do with some physical characteristic, or how you respond to a certain medication, or your risk for a certain disease, or whatever it might be. Tell me a little bit about Malika, whatever it is. Tell me about Francesca having to do with this. Tell me there's your story right there. And we use bioinformatics to understand it. And we're going to talk a little bit more about that. Okay, so DNA, although we think of DNA as this double helix, right? Think of it as two things, these base pairs, this is double helix. That's typically how we see it. I'm not going to talk much about this, but when it's sequenced, okay, when I'll talk more about sequencing, but when it gets sequenced, it typically focuses on just one of those two strands. So computationally, you're going to see your DNA represented just a long string of characters like this. A single strand. That's how you see it computationally. Because remember what I told you about base pairs? If this is an A, this has to be a T. And there has to be a T here. If this is C, that has to be a G, etc. But the point is, it's going to be represented computationally as a single strand. So you're just going to see, when we analyze things, it's going to be a single strand of bases or nucleotides or DNA sequence. Okay? Mental model. Okay, you don't have to remember any of this terminology, base pairs or adenine or any of these types. You don't necessarily, but I do want you to get the mental model. That's going to help you when we get to the bioinformatics part. Now, the only molecular biology I'm going to talk about this whole time here, and you don't even need to know about molecular biology, is the central dogma, because I think that's important to understand the central dogma of molecular biology, which talks about gene expression, right? Whether that gene gets expressed or not. Maybe those brown eyes won't get expressed in you. Maybe you got the blue ones and someone else got the brown ones, or you got the brown, someone else got whatever. Maybe it's not going to be expressed in you, or it's expressed differently. So the central dogma talks about gene expression. And gene expression happens in two stages. It is called transcription and translation. That's all I'm going to talk about molecular biology tonight. Okay. And DNA holds instructions for creating proteins. You're going to find that proteins is the whole of all this, for the most part, are going to be proteins. But DNA holds instructions for creating proteins. Okay. There's your DNA. Remember the base pairs and this double helix and all of that. But before making a protein, you guys, your DNA sequence is used as a template to create RNA, what we might call messenger RNA. We don't need to get that much into it, but you'll see that sometimes. But we want to create RNA through a process known as transcription, or it's transcribed. Its information is transcribed into this temporary working copy of instructions. I won't get into why DNA doesn't do it itself, why it needs to be transcribed, it has to do with the nucleus and some other stuff, which I won't get into here. All I want you to know is DNA hands its information over, a copy of its information, because you can't get to it. It protects its information. So it's going to give you a copy of its information to RNA. And so RNA is a temporary working copy. We also call it transcript. Okay. And then the instructions in RNA are read and translated into a specific protein. That process is called translation. Okay. And in reality, it doesn't really create a protein. It creates a chain of amino acids. You may not remember all this. I'm going to show you visually in a moment, but it really creates a particular specific amino acid. It's those chains. The chain of amino acid will create the protein that's desired, whatever its function is. Okay. So let's talk through this. Remember mental model. If you don't remember any of that, I want you to remember this. DNA is this four-letter alphabet that's telling that story of why does the elephant look like that, or why does that thing have a horn, or why is it that your hair is long and your sister's hair is short, or whatever it is. These are the letters that tell that story. So if we want to go transcribe from DNA to RNA, because remember, DNA has the story, but it doesn't directly create the protein. So DNA has the converter hand its information over to RNA. And so when it does that, RNA uses a slightly different alphabet. Those thymines get converted to uracils. You don't have to remember that, but U's will replace the T's when it gets transcribed into RNA. You don't even really have to remember that. You're going to see that when we get into Python in a moment, or in the code. You don't have to know any Python or coding, but I'm going to show you it. I want you to lean into this to know that you can do this. Okay, so this is what this DNA, this is what it would look like in the form of RNA. The T's get flipped to U's. Okay, now we have a strand of RNA. Now we need to translate it. RNA doesn't do us any good. We need a protein. So how are we going to do that? We need to translate it from RNA to a protein. Well, how does that happen? Well, you guys, you know what? It turns out that RNA is read as triplets, known as a codon. It reads this triplet that specifies a particular amino acid. And so here's what I want you to see. There are 20 amino acids for the most part. There's 20 different amino acids. And this is a lookup table to figure out what is the particular amino acid. But there are 20 different amino acids. And so here's how I think of it, you guys. We're translating from RNA's 4-letter alphabet to the protein's 20-letter alphabet. That's all this is doing. Okay, so we're translating from the genetic language of bases or nucleotides to the protein's 20-letter amino acids. And if you want to think of it this way, here's how I think of it, just to simplify it. It's like Google Translate. You're translating from one alphabet to another alphabet. That's all that's happening. This is one alphabet, A's, U's, G's, and all that. This is a different alphabet. It's a different language. And that's what translation does. Let me show you how it does. See this first nucleotide or base, which is an A? In this lookup table, it says, oh, go to the A quadrant because you see an A there. This is how it's read. And then it says, what's the next base or the next nucleotide? Oh, it's a U, so it goes there. What's the next one? It's a G. Oh, so it instructs for the addition of the amino acid methionine. You don't have to know what the amino acids are, but it says, oh, put that in the chain. Add that to my chain. That's what DNA told me to do. No, those are instructions I got from DNA. It says I need a methionine right there. Okay, I got you. Okay, now there's another thing I want you to see. There's something called stop codons. Stop codons tell it when the chain is finished. Oh, stop, oh, there's my protein. That's all I need. So a stop codon, which you'll see represented as asterisks, you're going to see that in a little bit. So just know it also codes for stop codon, telling it when the chain has finished. Okay, let's go to the next triplet or codon. Oh, it's a U. So it says go to the U quadrant. What's the next one? Oh, it's a C. And then the next one's a U. So it says, you know what? Can you add a serine amino acid to this chain of amino acids I'm putting together? And if we go ahead and translate the rest of them, that's what RNA is doing. It's translating from that 4-letter alphabet to this 20-letter alphabet, which are amino acids. Okay. And it's these amino acids that form a protein. Okay. So the sequence of amino acids determines the unique structure and function of a protein. Okay, so proteins are produced from chains of amino acids. And so we'll have all of these we'll have this amino acid chain that we're going to look at until it comes to a stop codon that says that's the end of the chain. I've composed the protein. Oh, there it is right there. Yep, that's what I want. That's what the DNA told me to create right there. There it is. That's the protein. And then it'll go on and read, and it goes to the next chain until it's, oh, there's a stop codon. Nope, there's my protein right there. Oh, that's it right there. That's what I was instructed to make, right? That's the protein that I need right there. Okay, so we look at all these sequences, and proteins differ from one another, mainly in the sequence of amino acids. Okay, you don't have to remember all this. You're going to see this in a moment. But I just want you to get a sense when you see this later. Oh, I told you what know what a protein is. It's not a thing. It's not one thing. It's a sequence of amino acids. Okay, we call it a protein, but there's all kinds of shapes and 3D, all these types of things that determine the shape and also the function of that amino acid. So a protein can be hundreds all the way through thousands of amino acids in length. Okay, so to sum this up, this protein producing sequence, remember that sequence we just read, those triplets, those codons, that's called a gene. That's a gene. A gene has the recipes for creating a protein. Okay, so a gene's going to say, read this codon, amino acid. All those amino acids are kind of chained together until it's finished. Says, oh, there's the protein that I need. So all the physical characteristics of your body are the result of thousands of different proteins that we call genes. Okay, so when we talk about proteins, if you're not familiar, I know you know the term protein, certainly maybe from nutrition and things like that. But proteins, you guys, perform most of the activities that keep the cell and the organism alive. Executing its specific functions, from a muscle to enzymes and hormones and antibodies. Just about everything is derived from proteins. There's other things, of course, water and fats. There's other things, but protein is so crucial, and we'll talk more about that, especially in terms of appearance. So let me look at this. DNA sequence determines this is the take home. DNA sequence, your genome, your DNA sequence determines the protein sequence, transcription to translation. Okay. The protein sequence determines the protein structure, which you may not know about, but there's this unique 3D structure that a protein forms. Okay. And then the protein structure determines the protein's function. So we go from DNA that we're going to read in your genome to the function of a certain protein. That's really what we're doing. I'm just breaking it down in terms of the transcription and translation. Okay? Now, let's talk about genotype and phenotype. Okay, just a second. Let me unblock my screen here. Just a second. Again, you guys, if you have questions, please feel free to unmute, or if you want to stick it in the chat, I'll try to respond if I'm not looking at my slides. I'll try to do that. If not, I certainly will respond by the end. Okay, let's talk about genotype and phenotype. Because when we talk about genotype, in bioinformatics particularly, because there's different ways to interpret genotype, but in bioinformatics, genotype primarily refers to the particular variant, or we also call it an allele, at a given position in the genome. Okay, so recall that the human genome is composed of two pairs of chromosomes, one from each parent. So we have two copies, right? And so at a particular location, you're going to have two different bases, or two different what we might call an allele. Okay, so here we might make a genotype call of TC at this position. A sibling might have two Ts at that position. Okay, and it's these different variants or alleles that can result in varying traits. Okay, why your little sister's taller than you are, or whatever it might be. Okay, it's these oftentimes these alleles or these differences here that can dictate what we're going to talk about next, and that is phenotype. And phenotype is an observed trait. So when we see a person with a given trait, their phenotype, we know that they carry a specific genetic sequence that makes that trait possible, their genotype. Okay, so in cancer, the features of the disease are the phenotype. Okay, but it's the mutations and variants within the individual which are the genotype. And a lot of times in research, we want to kind of link the genotypes and phenotypes together. In other words, what we are observing to what's going on in the genotype so that we can maybe improve our understanding of a disease or how, of a diagnosis, or understanding how a disease works. So we can see if what's going on in the genotype and how it's evidenced in the phenotype. Okay, let's talk about variation. So when we talk about variation, you guys, human DNA consists of about 3 billion base pairs, about 3.2 billion, but about 3 billion base pairs is the human DNA. And approximately 99.9% of those are identical in all people. Isn't that crazy? So if we all had our DNA sequenced, there's your DNA, and what's your DNA look like? Oh, here's mine. Oh, Francesca, let me see yours. Oh, Malika, what's yours? And so we're going to see everyone's DNA. It's going to look almost identical. All the 3 billion bases in one long string, if we could look at 3 billion things at a time. A machine can, but we can't. That's why we have bioinformatics. But if we could, we look at everyone's sequence, we say, isn't that incredible? I can't even tell a difference. And so, except for identical siblings, you guys, any two people have genomic sequences that differ at approximately 0.1% of the positions. Okay, so although most of our DNA looks the same, if we look closely, we'd see that there are some differences. Okay, and those differences explain why you and I look different, or behave differently, maybe, or why you don't get sick very often, or whatever it might be. And variations, you guys, at a single base pair. See, just one variation. See, these two base pairs are different. We were the same. Oh, why are we different there? A variation at a single base pair is called a SNP, single nucleotide polymorphism. Remember these bases, also known as nucleotide? Okay, so single nucleotide polymorphism. I'm going to talk more about that in a moment. Okay, and so we have a few million SNPs that differentiate. That's why we're different. We have a few million, because 0.1% of 3 billion is still a few million. So even though our DNA is largely the same, it is different at points. And so let's talk about SNPs, because this is what we're going to get into in a moment. A SNP, single nucleotide or base, or one of those letters, right, in your genome, A, C, G, and T, single nucleotide polymorphism. Okay, nucleotide, you know what that is, a base. And polymorphism just means many forms. A bunch of different variants that could be, there's mutations and all kinds of different things. So each of us has an accumulated collection of SNPs from our ancestors, whatever that means. And so if we were to look at a chromosome, let's say that this is a chromosome here, okay? Remember, you have 23 of them. 46, I should say, but 23 pairs of chromosomes, okay? So one base is inherited from mom, and one base is inherited from dad at this location. And just so you know, each pair of your chromosomes that you get from mom and dad, they contain the same genes. That is just incredible to me, that they have exactly the same genes, but not necessarily the same versions of those genes, what we call alleles. Okay? Everyone in your family might have brown eyes, but your mom carries a blue eye gene. Maybe. And so if you get that, depending on who your partner is, maybe you out of nowhere, the only kid in the family has blue eyes. How did that happen? Okay, so it depends what you're carrying, your genotype versus the phenotype, how it's expressed. And so remember, you get one from mom and one from dad. So if you were to read this right here, you would say, you know what? On chromosome 5, position 7, one parent gave you a T and one parent gave you a C. That's what we would say. That's how we would read that. That's how we would say what that allele or that variant is there. I'll talk more about that in a minute. Another thing I want you to know is that there's something called a reference allele. And a reference allele, in this case, we're going to say T is the reference allele. And the reference allele says it's the base that's found at this position in the reference genome. Okay, so for a given organism, we've gotten its sequence and we know what it should be at that position in the reference genome. So in the reference genome, there's a T at that location. That's the reference allele. Yep, we've sequenced a human before, and you know what? Turns out there's a T at that location. So that's the reference allele. It's the representative example of a species, or it's the standard for how we identify variations. That's the reference allele. Okay. There's also an alternative allele. It's any base that differs from the reference. That wasn't there on the reference genome. How in the world did you get a C there? Okay. So anything that's not in the reference genome at this position, it represents the variation or mutation at this position. Okay. So an individual with this SNP, single nucleotide polymorphism, different forms, different variants, would have a genotype of TC. That's what you'd say. On chromosome 5, at position 7, their genotype is TC. So we'd say they're heterozygous. Hetero means two different, heterozygous for two different bases, T and C. Another person may come along and maybe their genotype is TT. They're homozygous for the reference base. That's what the reference genome had. And turns out you have two T's there. Or a person could be could have the genotype CC, and they would be homozygous for the alternative base. Okay, we didn't see anything like this on that reference genome. This person has two C's at that position. Okay, these are polymorphisms. Okay, and polymorphisms are what make individuals different from one another. Okay, and just so you know, most traits that we have are SNPs at multiple locations, not just one. Typically, we'll have multiple, like in a gene, there'll be multiple locations where we have SNPs. Okay, well, I'm going to look at SNP at one given position. Okay, so it's these variants, you guys, that tell your story, that tell about your ancestry, or tell, influence your disease risk, or predict aspects of how you look, such as your characteristic hair and eye color. See, SNPs that determine that are, you inherit a gene or genes that make proteins causing you to look like you do. Okay, or height. Okay, these variants or these SNPs tell a cell how to function and what traits to express. That's why we're looking at these things. In other words, we take a bunch of tall people, and what pattern is there in their genome that doesn't exist in other people? What's making them tall in their genome? We can do studies like that. Okay, what is turned on, or what SNP, what variant is in that person that doesn't exist in other people who aren't so tall? Okay, or susceptibility to a disease. It's largely inherited. And just so you know, many SNPs have little consequences. You'll have SNPs that say, oh, that doesn't do anything. Oh, this one doesn't do anything. They apparently don't have any impact that this is different and different and different. But sometimes, especially if they're homozygous at a particular position, they could be lethal. And so it's important to understand that and try to link those relationships if we can. And so by examining the relationship between physical traits, phenotype, and the genome, your genotype, we can develop a personalized medical treatment based on the individual's genetic makeup that we call precision medicine. So such as a risk for disease or an indication for specific medication or whatever it might be. Okay, let me take you over to a notebook. You don't have to know any coding. But hopefully with your mental model, you're going to get what I'm going to talk about next. Some of you will know if you're going through Stanford Data Ocean, you've gone through maybe some of the R, the Python. But if not, I'm not even talking about coding. But I do want to show you really quick here. Okay, so let me get over here. So if you were doing this, you're going to get some sequence. Oftentimes, you'll see a genomic sequence in a FASTA file like this. Okay, so you'll have some DNA sequence, and this is what it looks like. You're going to say, what in the world is this? Just a bunch of G's, C's, C's. I'm a human. I have finite. I can't make any sense of that. So you're going to have to use bioinformatics to try to make some sense of that. You're going to get this thing, you say, I don't even know what this is. Or I'd like to learn a little bit more about it. Okay, this is some data. It could be a genome, maybe it's a protein. I'm not sure. But we're going to try to identify what this thing is. Okay, so if you were looking at this, you might want to know how long it is. And it's 48,502 bases long if you're just kind of playing around. There's a thing that we can do, which is called a blast search. And a blast is a database that can align your sequence with some genome. Where does this fit on some genome? That, in other words, I need to, the order of these bases, right? What do they align with? Whether it's a given organism or protein, and we'll talk about that. So you can use a blast search. It's called Basic Local Alignment Search Tool. And we're going to do a search here for nucleotides. We're going to call these nucleotides. I mean, they are nucleotides, but if they were amino acids, we would do a protein search. But these are nucleotides. So we're going to do a search to say, what genome does this align? You know, all these bases, do they align with any known genome out there? So when we do this search, it turns out that this thing right here, let me show you where the alignment is right here. It'll return a bunch of things, and it will show you how much confidence it is. But it turns out that the best matches from the bacterium E. coli is what it tells you. So you can see, it will tell you what that matches with. Okay, and it tells the complete genome is about 4.6 million bases long, and you can see that it's nearly a perfect match. That's what it aligns with. Okay, so even though you can't comprehend billions of base pairs when you use your bioinformatics skills, you can with these databases. People have come long before you and done a lot of research and have these databases and tools that we can use. This happens to be called BioPython. It's Python having to do with biological data sets. Okay, give you another example here. Here again, you have some DNA sample. Okay, so here we have a DNA sample. Let's say you just want to do a little analysis because you have coding skills and you have strong domain knowledge. You know your domain, whether you're doing cancer research or you're doing something having to do with, the growth of children in whatever it is. You know your research, you know your domain. So you have this sequence right here. So you want to do a little bit of an analysis. So you have at some point, you may have your Python skills. So you might say, what's the length of this sequence? Turns out it's 29,903 bases long. You say, oh, okay, that's interesting. Make this a little bigger here. Oh, okay, that's interesting. So then you may want to count the nucleotides, remember? You don't have to know what those are. You just know there's A, C's, G's, and T's, even if you don't know what they mean. Okay, you just know, I know that there's this 4-letter alphabet that the DNA speaks in, and I want to speak that language. So A, C's, G's, and T's, let me count them. And if you count them and you have domain knowledge, you can see what the frequency is of different bases. That might mean something to you. This many A's, C's, G's, and T's. And there's something called GC content, which I won't get into, but sometimes the relative proportions of those bases or nucleotides can be meaningful. And so this says the GC content, or the proportion of the A, C's, G's, and T's that's either a C or G, is 38%, and that might mean something to you. So again, having the skills to do things like this as you're analyzing your genome or whatever it might be can be useful. Now we have this this DNA. Let's transcribe it to RNA. Okay, so if you want to transcribe to RNA, remember those T's just get flipped to U's. Now, let me go back up here. I'm sorry, you guys, for all the scrolling. See how this says A, T, T, A, blah, 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 blah. I'm just going to look at the first 50 bases just so you know, make it easier for you to see. Okay, so you see A, T, T, A. I'm just looking at the first 50. So here's the first 50 DNA sequences. And when we convert them to RNA, you see how those get flipped to uracil or U's. Right, that's all you need to know. You don't need to go anymore. You don't need to know necessarily about molecules. And I mean, unless you need to, right now, you don't need to know that because I think you at least get the alphabet and what's going on. Okay, so I know this is RNA. It's just a temporary holding place for the instructions. I can't do anything with RNA. There's something that it needs to do next. And that means it needs to translate to amino acids. So if I want to see what the protein is in this DNA, I have the skills to do that. I know how to do that. And so what we do now is we're going to convert these amino acids, this RNA strand right here to amino acids. And so remember what it does? It will take that codon or that triplet, and it just reads it just like I did. Okay, so it just reads it and says, oh, here's this amino acid and there's that amino acid. This is a stop codon. It says, oh, stop, there's the protein. It'll go on and read some more. Oh, there's the protein, stop. And this is what it's doing. And so I asked, by the way, in my genome, how many amino acids are there? Oh, 9,967. Okay. And then I might say, what are the top 10 amino acids? Oh, a lot of leucine, some serine, and all these other things, if that means something to you at some point in your career, whatever, you might understand that. And so you certainly could visualize them. Visualize my top 10 or top 50 or top 100, I mean, not 100, but top 10 or top 20, whatever it is, amino acids that I might want to do to see if that's important. Again, when you have domain knowledge, that might mean something to you. Okay. And then after that, we want to look at proteins because that's ultimately what this is all about. Remember, proteins are split on that stop codon, that asterisk. So here's a bunch of proteins, but that's not going to make any sense to you. That's why you have coding skills. So I said, how many proteins are in this DNA sequence? Oh, 775. Huh. Okay. Can you show me some of them? Yeah, here are the first 10. It shows me the length of them. I wanted to know the length of them. I said, okay, can you show me the top 10 by length? Yeah, the longest one had 2,701 amino acids. The next one had 290, blah, 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 blah. Okay, I said, and typically a functional protein is typically longer than 20 amino acids long. So I just said, how many are at least 20 amino acids long? Don't worry about the coding. And it said 80. And so I'm calling those functional proteins, at least 20 amino acids long. Okay, if you have domain knowledge, you'll have your own way. Okay, so if I were to summarize this analysis, I would say the sequence length is 29,903 base pairs. There's a high amount of leucine and serine. There are 80 functional proteins, and the largest protein is 2,701 amino acids in length. And again, if you have domain knowledge, that'll mean something to you. But I took another step. I said, well, I still don't know much about this thing. I mean, what's the function? What does it do? And so I took the largest protein right here. There's 2,701 amino acids. I don't even know what it is. What in the world is it? I mean, I'm looking at it, but what does that even mean? What does it do? And so I took that and I saved it out. Okay, I just called it largest protein, saved it out as a FASTA file, which is a common way to do it. And then I went and did my blast search. But instead of looking for a nucleotide alignment, I did a blast search looking for alignment for proteins, because I know that's a protein. Remember, I'm in a different alphabet now. I'm no longer in that 4-letter alphabet. I'm in the 20-letter alphabet of amino acids. So I'm going to say, I want to do a protein search. Okay, so I'm going to do a protein search and say, what does this tell me about this thing? And so when it did this sequence, look what it does. This is what it brought back to me. And it tells me, first of all, it found a very strong match to a protein associated with coronavirus. It says, you know what, you're dealing with coronavirus, COVID-19. And it tells me that this protein is the enzyme right here. This protein is the enzyme that helps the virus replicate its RNA, which is crucial, important for its reproduction. It tells me that. And it tells me, as you look right here, that these sequences are almost identical. It aligns them for me. See, it says these sequences are almost identical. Not identical. There's a polymorphism here. These are different there. Those are different there. Most of these are all the same. And then this last one is different. So, but in general, it says over 926 bases, it's almost identical. Okay, so now it's confirmed the identity and the function of that protein. That maybe that's what I know I'm dealing with COVID-19. I know it has to do with it replicating itself. And I know that it matches extremely well with that. So I can be fairly confident in terms of the identity and the function of that protein that I'm dealing with. Okay, I know this can be a lot for some of you who don't know Python or anything. Don't worry about it. Python's so human readable, you guys. It's not that difficult. It's more coming up with a brilliant question and getting a data set to play around with. That's when you take the next step. Okay, let me get away from that. Now, the next thing I want to talk with you guys about here, let me see how we're doing time-wise. Okay, is I want to talk about machine learning, you guys. That's where we are. Machine learning is magic. You guys, I do this all day, every day, and it's still magic to me, machine learning. It's incredible. And so we want to talk about machine learning with genomic data. But remember, you can't see 3 billion anything. The machine can see all of it, and can learn. You have to just ask a brilliant question and get a great data set. Whatever it is that you're working on, you want to say, what would change the world, change the market, change the research? You have to come up with a great question and let the machine do what it does well, but you have to have machine learning skills. Okay. The first talk that we had was on machine learning. The second talk that we had was on data preparation. I'm going to review those really briefly so we can talk about it with genomic data. Okay. So the first thing is, here we have a data set. This is what many of you are probably more commonly have worked with, is structured data in Excel or whatever, Google Sheet or whatever. And here's a data set. This is structured data that fits in rows and columns. And it, we want to predict heart disease, let's say. Okay, I want to give you a mental model so you get it, so we can go to genomic data. Oh, I totally see it. Okay, so we have a patient's age. We have a patient's sex, their type of chest pain they're presenting, their resting blood pressure, their cholesterol, their max heart rate, whether they have angina, and then whether they have heart disease or not. The first question the machine's going to ask you, what question are you asking me to learn, to predict? Oh, yeah, I want you to tell me if a patient has heart disease. This is the target variable or the answer key, because the machine's going to say, well, do you have examples of the world? Do you have examples of a person with heart disease and someone without heart disease? You're going to say, oh, yeah, of course. Yeah, see this person right here. This is heart disease, and this is heart disease, and this is not heart disease. That's how the machine's going to learn. That's your answer key. You put that off to the side. That's your target variable. Everything else we call features. That's the machine's knowledge of the world. Machine knows nothing about the world but what you tell them those features there. It's trusting you. The only reason the machine needs you because you live in the world and you're human. The machine doesn't need you. You can't look at a billion. You don't know anything other than that you know the domain and you live in the world. You can describe the world to the machine. So you need to get to describing the world. We call these features, you may know them as columns or independent variables or whatever, but we call these features in data science. And so the goal is to learn to predict the target given the features. That's what machine learning is going to do. So now machine learning says, look, I'm not human. I don't live in the world. I don't understand any of this. Why are you giving me that? Just give me the numbers. And sitting between those examples of the world is going to be some machine learning algorithm. And the machine's going to say, oh, okay, let me see here, Malika. Okay, let me see here. So you're saying this is what a person with heart disease looks like, huh? And this one too? On this person, oh, okay. It's trying to find a pattern. It's just a sequence of information about the world. And this algorithm is brilliant, incredible how these algorithms learn from data. And it's saying, oh, okay, so this person has heart disease, huh? Okay, and that person doesn't, and that person does, and that person does. Okay, it's trying to learn a pattern. Okay, and once it does that, it says, you know what? Just give me the numbers. Put the features in an X, put the target in a Y. And now you're saying, machine, can you see what heart disease looks like now? Can you see what I'm talking about? Can you tell if I were to give you the patient's age? Blah, 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 could you tell me whether they have heart disease or not? That's what we're trying to do with machine learning, because the machine can see all of it. Okay, machine can see all 200,000 patients all the way back to the 1970s. You've been collecting this in your hospital forever. You have all this data. What are we going to do with it? Well, I have machine learning skills. Machine can answer anything I ask it if I give it enough examples and tell it the truth about the world, organize it in a way that truly reflects the world. So let's talk now about model training. With model training, this is you. This is why the machine needs you to collect examples of your problem. We call it data. The machine doesn't know what data is. The machine just calls it examples of the world. Give me examples of someone with heart disease. Give me examples of someone who gets the promotion, or examples of someone with benign versus malignant cancer. Just give me examples. That's your job. You can call it data if you want. Machine says, just give me examples of your world. And that's your job. If it's genome, give me examples of that person who, whatever this condition, this rare condition, give me an example of their DNA, of their genome. You let me figure out what might be different from the reference genome or from these other patients or from how they were 6 months ago or whatever it might be. Okay, or a sibling, a family member, or whatever it might be. So this is data. This is you. There's some algorithm here. You two work together to form a model. And the model knows nothing, so it makes random prediction. But you're smart enough to give it that answer key. And now the machine, what we call supervised learning or supervised machine learning, the machine can say, oh, that person doesn't have heart disease. Oh, and this one doesn't either. And this one does. And the machine can go back and adjust this. It will modify itself iteratively till it gets a little better. It's going to say, you know what? I think I can see when someone has heart disease now. Nope, I got it now. I got it now. We call that a trained model. It's the machine that's doing all of it. As soon as you've given it enough examples, you've given an answer key, and you've asked a brilliant question. Okay, now the last thing that you need to do for the machine is data preparation. This is how we interpret the world for the machine. The machine doesn't know how to interpret the data. You'll see what I mean by interpreting it. So let's talk about review data type. We've talked about this before. If you have numbers in your data, the machine gets numbers. 365 days a year, someone's IQ, SAT score, those are integers. Machine says, I got this, don't worry about it, I got numbers. Or continuous values, pi, GPA, decimals, I get that. If you have numbers, machine understands numbers. But sometimes you have categorical variables, like gender, or race, or position in a company, or education level, or those are categorical variables. Machine says, I don't know what to do with that. I don't live in your world. Those are nominal. I don't know what they mean. They mean nothing to me. So you have to communicate those concepts numerically. And then lastly, if there's some order to your world, I need to know that. First, second, third place. Freshman, sophomore, junior, senior. Oh, there's an order to that? They're not just random names? Oh, there's an order. Oh, so you go from, oh, okay, tell me that about your world. I need to know that. Okay, 3-star, 4-star, 5-star restaurant. Okay, tell if there's some order to something, I need to know that. Okay, so here we have a data set. And let's say HR wants to predict whether, if things get a little tricky in the world, if we're going to retain someone or furlough them. Okay, HR wants to create this model. So here we have our data. Remember the machine's knowledge of the world. We have gender of an employee, age, their race, their tenure, how long they've been with the organization or the company, their position in the company, their average pay rate, and whether they've been retained or not. The first thing the machine's going to ask you What question are you asking me to predict? What do you want me to learn to predict? You're going to say, oh, yeah, yeah, yeah. I want you to predict whether someone's going to be retained. Because for all the machine knows, they want you, maybe you wanted to predict someone's gender. From machine doesn't know. So you have to tell, oh, yeah, I need you to predict whether a person will be retained or not. Machine's going to say, well, do you have examples? Oh, yeah, yeah. See, in the past, this person, these two people weren't retained. They were furloughed. These two people, blah, blah, blah. See, machine, can you see what I'm saying? Can you see the pattern? That's what you're trying to get the machine to do. Everything else are features. And this is what we need to prepare so that the machine can understand our world. These features, this is all the machine knows about the world. If there's something else the machine needs to know, or that you would like to know about this problem, you better go run and get it from the machine. Machine's trusting you. This is all I need to know about your world, right? Okay, well, let's go. So the next thing you need to do, once you've done that, you've settled on that, we need to transform the data because machines want numbers. Okay, machines need numbers. So for this HR data set, we need to transfer your knowledge of the world, your knowledge of the problem. So first of all, gender looks to be binary. It may not always be a binary problem. So you need to interpret this for the machine. So this looks to be binary, so I'm just going to arbitrarily make male 0 and females 1. Okay, machine doesn't know what male and female is. Machine just says this thing or not. It's like a light switch, on or off. You're this thing or you're not this thing, whatever this thing is. Machine doesn't care. You're saying it's binary. It could be anything. It's just binary. Okay, then we have age. Oop, those are numbers, totally number. Machine says, move on. I got it from here. Yep, 44 is necessarily twice as big as 22. Those are numbers. Let's go. So what about race? Well, the problem that people do with the race that some of you may remember, because you know you need numbers. And so you're going to show up with the team. You're going to say, you know what I did? I just converted them to numbers alphabetically. Yeah, that's what I did. So this is Asian and this is Black. But the problem is we don't think of race as ordinal. We don't think of race as being in an order unless you do. But we don't think of, you're telling the machine that one race is better than another. Yeah, machine, this is the best race, and this is the next best race, and this is the worst race, or whichever order. We don't think of race as ordinals. You have to say, oh, machine, you know what? No, 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 no. I didn't mean to tell you that. I'm sorry. So what I need to do instead, I need to do what we call one-hot encoding. There's one thing that we can do is when we have data that's not ordinal. Okay? So when we do this, instead of giving it race column gets dropped. And each category gets its own column. So now if you're Hispanic, you get a 1. Everything else must be off or false. Okay, if you're Asian, you get a 1. Everything else must be zeros. If you're Black, you get a 1. Everything else must be 0. So you're saying, machine, just to let you know, I'm sorry, I didn't mean to put them in order. No, 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 no. That's not how our world works. The person just incidentally this race, and I'm not making any claims about order or degree difference among the races. If you can find a pattern, please find it. But no, I can't put these in order. There is no order. We don't see them as being in order. That's what you're telling the machine here. Okay, now tenure, we think of tenure just like age. So there's some order if you want to. These are truly numbers. There's some magnitude difference here. Those are good. Now position, it determines how you interpret position. If this is an entry-level position, then you go to the next position, you go all the way to 3, then 4 is a senior level position. I can put those in order. I think that's ordinal. Yeah, you go from a 1 level, then you go to your 2, and you might make it to a 4 one day. Yeah, I can put those in order. But if these aren't ordered, if these are the same position, but maybe they work remotely just in different cities. This person's in San Francisco, this person's in Boston, this person's in LA, this person's in Phoenix. So I can't say that one is better than another. Or these are just different buildings on campus. This person works in building 1, this person's in building 2, or whatever. I can't put them in order, machine. I'm sorry. Yep, I shouldn't have put them this way. These are just designations, but one is not necessarily better than the other. So I need to one-hot code. If you're position 1, you get a 1, everything else must be off. Position 2, you get a 1 there. Position 4, you get a 1 there, etc. Okay, so keep that in mind when we get to genomics. You're going to see this in genomics in a moment. Okay, that's why I'm taking the time to go back through this. We talked about much of this in our previous lecture. Pay rate. Okay, so pay rate here, these are numbers. Machine says, I'm good, move on. Those are numbers. No, 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 those are numbers. And then retain, this looks binary. So it says you can convert no to 0 and yes to 1. Okay, now you've prepared your data. This is the only reason the machine needs you. You have to interpret what those numbers mean. The machine needs you to interpret the world. Once you've done that, the machine says, this is for human eyes. I don't understand what any of that means. I don't read. Just give me the numbers. Put the features into X, put the target into Y, and some machine learning algorithm is going to sit in between them. Machine's going to try to learn from X to predict Y. See these series of numbers? You're going to see this in genomics in a minute. It's just this row of numbers. The machine, because it's so good, it's going to look at this row of numbers and say, okay, so you're telling me when I see this, I should predict close to 0, huh? Okay, and when I see this, I should predict close to 0. Oh, now this one's a one, huh? And this one's a one. Let me look at it a little bit. Let me see if I can. And that's what the machine's going to try to do. It's to learn the pattern in the data. Machines are brilliant if you give them enough examples of your world. Okay, and if the problem is solvable. Okay, if humans can solve it, if we can tell, if it's solvable, then machines are actually much better than you are. Okay. Um, Lastly, we want to talk about genomics, you guys. I did all that to get to this because we've talked, for many of you, we've talked about that, some of that before. But I did that to make sure everyone's at the same point because this is why you're here, you guys. Hopefully, you have these skills. Machine learning is brilliant, just brilliant what we can do today, whether it's deep learning, machine learning, these large language models. You want machine learning skills if you're really serious. And not that you can't do it without it, but man, it just puts you on a totally different level, the things that you can do. Because you just have to come up with examples and a great question, let the machine do all the stuff that humans can't do. And you'll see what I mean in a moment. Now, the problem that I came up with, you don't have to know anything about this, but there's something, this is just a project that I chose to do. You don't have to know what this means, so don't get thrown off. I want the machine to predict whether there's a transcription factor binding site in my data. And all I want you to know about this, to give you a mental model, a transcription factor can influence gene expression. In other words, if it binds to a certain location, binding site, it can determine whether a gene gets expressed or not. So it's looking for these binding sites. And so these binding sites are just a sequence of nucleotides, it's a sequence of bases, but it's a certain pattern that it's looking for. Aren't machines great at that? Looking for patterns? A machine, can you learn the pattern that have to be the transcription factor binding site so I can predict gene expression in this DNA that I'm looking at? Okay, so again, it could be any problem. You might want to say, can you look for patterns that are that relate sex and race to this form of cancer? I'm giving you a bunch of examples. Is there any pattern in the genome or in this sequence or whatever it is? You'll come up with a brilliant question. It's all just patterns to the machine. Machine doesn't know a transcription factor from heart disease, from anything else, from whether you're going to be furloughed or not. It doesn't know anything. They're just a series of numbers to the machine. But this is what I'm going to do because I know it's just looking for a pattern. That's what a transcription binding site does. Look for a pattern in those nucleotides, those bases that it can bind to, and it can influence gene expression. So let's get to what you guys know now. Here you happen to have the data set. And this data set right here is, this is just a genome sequence. I'm just showing you a short little to make it easy to see. But this is base 1, base 2, base 3, base 4. So it's just a DNA sequence. Okay. And then we have whether something's a transcription factor binding site. That's how you have to set your problem up. And the machine's going to say to you, what question do you want me to answer? What do I need to learn to predict? Oh, yeah. I want you to, if you can tell me whether this is a transcription factor binding site. That's your target variable. Maybe you want the machine to predict the base that's in position 3. The machine doesn't know what you want it to predict. So say, oh, no, here's my answer key. The machine's going to say, do you have examples? Oh, yeah. See, this is a transcription binding site, and this is a binding site, and this is blah, blah, blah. Machine, can you see it? Okay, hope you get the mental model of machine learning. Machine learning is not you. Your job is to collect examples of the world. Hopefully, if it's supervised learning, you have an answer key, and get out of the way. Okay, just ask a great question, let the machine do it. Okay, and have some level of threshold that would be successful. How accurate does it need to be? Some things, 25% accuracy would change the world. And other things, you might have to be 95, 99% accurate even matter. So really, those are the things you'll have to decide, but machine learning is not you. Okay, now once you've done that, remember everything else are the features. This is your DNA sequence. This is what you've collected from a patient, or maybe it could be a protein sample. It could be anything, but it's a sequence of some type. In this case, it happens to be nucleotides or bases, but it could be amino acids. It could be whatever you want it to be. Okay, this is the machine's knowledge of the world. Okay, remember now, we took all this time because the machine says you need to transform the data because machines want numbers. Those aren't numbers. You can't give the machine those bases. Machines know what to do with those. You can't say, well, you know what I'm going to do? I'm going to give A a 1, I'll give the C, I'll turn that to 2, I'll give the G a 3, the T to 4, because those bases, we don't think of them as being ordinal like race. One thing of one base is being better than another. We can't put the bases in order. So you're probably going to have to one-hot encode them. It's one of the ways we do it. So the way we one-hot encode these bases, A, C, G, and T, hopefully you guys can see it. Just like this. If you're an A, you get a 1 in this first column, everything else must be off. If you're a C, you get a 1 in the second column, everything else must be off. If you're a G, you get a 1 in the third column, everything else must be off. If you're a T, you get a 1 in the fourth column, everything else must be off. That's how you're going to give your data to machine learning. Okay? So let's go back to our data set. You're smart enough to know, ooh, I'm excited about this data set. Oh, Tim, you know what? One thing really quick. Oh, no, we need to transform this really quick before we give it to the machine learning model. Oh, yeah, I'm glad I remembered that. Yeah, so we need to take this first base. And yeah, I'll just one hot encode it real quick. There you go. Because remember, look, this first happened to be CAT. Remember, when you one hot encode it, CAT. That's one hot encoded. That's how the machine will see it. Now, you're human. You would rather see it the other way, but that's not how the machine works. And then base two, you have A, and C. So you have A, and C. It's one hot encoded. And then base three, you're going to one hot encode. And base four, you're going to one hot encode. And the machine says, ooh, looking good. I like this. Okay, now what do you ask me to predict again? Oh, yeah, I need you to predict transcription factor binding sites. And machine says, remember, I need numbers. Yep, you're right. Let me change yes to 1 and no to 0. Then the machine says, okay, looks good to me. Give me the numbers. Put the features in X, put the target in Y, and sitting between your examples of your genome, you're going to say, machine, can you see when there's a transcription factor binding site in the sequence or whatever brilliant question you've asked the machine? Can you see that SNP that predicts how the person will respond to this medication or whether they'll be alive in 10 years or whatever it might be that you're trying to predict? Okay, now let's go back over. And you're going to see why your Python skills will really amplify you. Here we have a data set. Same thing I've already told you. We want to predict there's a transcription factor binding site. And to make it easy for you, I just kind of simulated some data here to make them 50 bases long so that you could see them so it didn't go on forever because I wanted it to be on the screen. So I have 2,000 DNA sequences, 50 bases long. And that's what they look like. I'm just showing you the first 5. Okay, and if I preview this first one right here, CCGA, whatever, this is it. This may be a sequence that you have, although it could be 10,000 bases long or a million bases long, whatever it is. But this is going to be the first sample that you have. Oh, okay, so that's a patient, huh? Or that is whatever it is that you're doing. This is it. Okay, well, before I can do any machine learning, you know you're going to have to one hot encode that. And remember how we one hot encode? This is what an A looks like, a C, a T, or G. So if we had a sequence that was CCGAGT, this is how you have to convert it for the machine. Okay, see, this is a C is in the second position. C. Let me make this bigger. I don't know if maybe you guys can't see that well. C, G, A, G, T. That's what it looks like to the machine. Then it gets flattened out. Remember how we had heart rate data or furlough or retained data? That first row is just a series of numbers. Well, that's what this is. Machine doesn't know if this one means sex of the person or whether that means they were promoted or whether they have a graduate degree. Machine doesn't know. It's just a series of numbers, and the machines are good at looking at patterns. Okay, this is now what you see as a human with all of your molecular biology background. You see this way, the machine says, no, 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 no. This is how I see that. Okay, and so this sequence right here that you see, when it's one hot encoded, it looks like this. Okay, C, G, A, G, etc. This is what that first 50, it ends up being 200 numbers long. And when it gets flattened out, this is what it looks like. Now, it wraps around just so it can stay on the screen, but if not, it would be 200 numbers long because just 50 bases. Think about if you had a million bases or something. So this is what that first sequence looks like. So let me go back up here. Sorry for all the scrolling. But with this to you right here, because you've done your homework, you have your Python skills, looks like this to the machine. So let me process all 2,000 bases. When I process all 2,000 bases, whoops, sorry, you guys. When I process all 2,000 bases, here's the first sample. Whoops, I'm sorry, no. Here's the first sample. Here's the second sample. Here's the third sample, fourth, all the way down to 2,000 of them. Okay, so once we do that, here are the labels. It says that first sample, remember that first sample I gave you? Yeah, turns out it has a transcription factor binding site. It does, yeah. And the next one, that second one I showed you, yeah, it doesn't have one. And then the next 3, they do, etc. We do that for 2,000 of them. And when you give that to the machine, you're just going to choose an algorithm because machine learning is not you. You don't necessarily have to know the algorithms. But logistic regression says, you know what? I'm 77% accurate. From that DNA sequences you gave me, yeah, those 2,000 samples, yep, I'm 77% accurate predicting whether there's a transcription binding site. Or a better way to say it, just for mental model, if there's that pattern that you mentioned to me. If you forget that, forget it. It could be any pattern. Like say, it could be cancer. Could be whether that basketball guy is going to be tall when he becomes an adult, or she, whatever it is you've asked me to learn. I've looked for that pattern that you've taught me about. Okay, I'm 77% accurate in predicting that pattern exists in the data you gave me. Support vector machine. I'm 98% accurate. K-nearest neighbors algorithm. I'm 85% accurate. Each algorithm is different. There's no one best algorithm. They're all different. In the next project, it may be a different algorithm. Okay, so hopefully you can see the value of machine learning, because as you come up with examples, there's some pattern in that sequence, those SNPs, all those things that we look for. Okay, so lastly, we're almost at about an hour, you guys. I want to wind down. I would like to finish within an hour. So the last thing that we're going to do here, this is from the module on Stanford Data Ocean. So if you're working through Stanford Data Ocean, you'll come to this Introduction to Genomics module. So let me just say this real quick, and then we'll wind down here. In this module, it takes you through some, now you have a good mental model. Hopefully you guys get this now if you didn't before. You'll see some of these things, whether you know the terminology or not. Okay, this variant calls and all that's just a variant, just a SNP. So let me show you right here. And when you look at the first part of this module, it shows you this thing from 1,000 Genome Project. It shows you different SNPs. It says chromosome 19, show you the position on the chromosome. Remember the reference base? There's a C on the reference genome. Then what are the variants? What are the alternative bases? Where there's some A's that show up sometimes too. That's not on the reference genome, but there's some A's that the researchers will show up at that position. And then this shows you what is the prevalence in different populations. Okay, this shows you as you read much of what I've told you here, and then we'll show you some of the population. So if you want to say, oh, what's the prevalence of that in East Asians or in the African population? Maybe you want to know that. Or maybe in certain types of patients. What's the survivability of that for those who are Black and, have some condition and they're over 65 or whatever. You'll have some combination of things that you're going to want to know about the world. Okay, remember SNPs? Well, I'm going to give you this site in a minute. SNPedia allows you to look at SNPs because there's a lot of research done, and it will tell you certain SNPs that the research says. For example, this SNP right here is highly associated with empathy. This SNP right here is associated with a dislike for cilantro, sleep disorder, skin cancer. There's a bunch of SNPs. And here's what the SNP will give you. Here's the chromosome. Here's the start and end position. Here's the reference genome base. And here's the alternative. And we'll tell you the prevalence in different populations. Okay. This one right here, this cosmic database allows you to look for SNPs and mutations have to do with cancer, if that is your research. And then lastly, you have a variant-based annotation. Variant-based annotation says, you know what, in my sequence, there's a variant that I've noticed, or a SNP, or a few SNPs, or whatever it is, and you want to do a search for them. You want to find something that matches them so that you get information about your SNP. What's the functional impact of this SNP? What's the disease association with this SNP? Or what is the prevalence in a given population that I'm working with? Okay, so variant-based means it's the other way. I have a SNP that I'm observing, and I want to search a database that may match this SNP to say, has anyone identified this SNP in their data set? And if so, what's it related to? I'd like to know a little bit about this. Okay, and then lastly, let me go over to SNPedia, and I'll give you guys this, and then we'll wind down here. SNPedia, you have an FY, in fact, let me put this in the chat for you guys, just so if anyone wants to follow along, and then we'll wind down here. So if you want to follow along with what I'm doing, this is SNPedia. So if you're interested in SNPs, just out of curiosity, or if you're just getting your career is just getting started, and you're thinking, I really want to do this, or maybe you're in the middle of your research and you're really interested in certain SNPs, and you can look at it by genes, genotypes, medical conditions, medicines, topics. So for example, this first one, muscle performance. If you click on muscle performance, it's going to take you to this page right here. And it tells you on that SNP, if there's two C's, maybe you get one from mom and one from dad. If you have two C's on that SNP, it's related to better muscle performance, likely a sprinter. And just looking at all the different research that's been done. Okay? So it's kind of organizing a lot of these research studies on different samples. This says if there's a C and a T there, if it's heterozygous there, it's still likely to be, it's a mix, but it's still likely to be a sprinter. If you have two T's there, impaired muscle performance, likely an endurance athlete. Now, if you click on the database for that SNP here, take you to NIH, and this is where it will take you. And it says Homo sapiens, chromosome 11, which is what this was right here. Where is that? Chromosome 11. It tells you the position on that chromosome. Okay, remember the base location. And so it says Homo sapiens, chromosome 11, there's the position. And it tells you in the general population, from all these studies that it's getting this from, here's the total sample size of the studies, right? And it says the reference allele, the allele that's in the reference genome is a C, 57% of the time. And the alternative allele shows up 43% of the time. Okay? It doesn't have to be dominant and recessive necessarily, but it just shows you proportions. Okay? And if you look here for those who are European descent, they're very similar to just the general population. But look at those from African descent. 82, 89% of Cs. And if you look at any 100-meter final in the Olympics, you may see a lot of people from African descent, at least for certain parts of Africa. Some other parts of Africa have a lot of distance athletes also. But the point is, this can really tell you some things. So as you go here, look, if you're from Latin America, 34%. You might be more likely to be a distance athlete. Look, South Asia, 42%. So I love looking at these SNPs because it takes all this research to say, what's the SNP? You know, we're looking at these different, these genome-wide studies. The other thing here, let me show you one other one, you guys. If you go here to red hair, if you click on red hair, it says that for red hair, if you have two Cs, if the SNP there or the allele there is two Cs, that's normal risk because red hair is rare. Okay. If you have a C and a T there, it shows you other things that you're a carrier of it, but you're also at risk for melanoma. If you have two Ts, it says you have an increased response to anesthetic, 13 or 20 times higher likelihood of red hair and an increased risk of melanoma. So it gives you all this information. So 2C is what's going to be on the reference genome. So when we click on the database here, when we go to NIH, when we look at it here, we can see again, Homo sapien, chromosome 16 at that position. And the general population, about 93% of people don't have that red hair allele, just only 6.5%. In Europeans, about 7%. And look as you go to African descent, some don't have any. 100% of the reference allele. So very rarely would, if at all, would be able to have red hair. Now, if you go down to other people like here, Latin American, 2%, others 5%. So I won't go through this, but it can be very fascinating, you guys, as you look at some of these SNPs, if you're interested as a layperson. These are some that they promote on their website here if you're interested, but you can also click here at a bunch of SNPs. Or if you do research, you see a research paper. This is an ID for a SNP. So if you do a research paper, you may see that. You can kind of match it up. Okay. So with that, you guys, I'm going to stick around. I've gone longer than I wanted to, about 6 minutes longer. But if you have questions, I'm going to hang out. Feel free to stick around with me and ask questions. Or if you want to poke around at that SNPpedia site, then I'm going to stick around for a while. So thanks, you guys. Really appreciate your attention.